out of 20 of these. We also did in silico analysis to see if the primers and probes that we were utilizing would cross-react with any of the common coronaviruses, and then some of the more severe coronaviruses, SARS and MERS, and also respiratory pathogens, both viruses, bacteria, and fungi that live in the respiratory tract. And indeed, our primers and probes did not detect these by in silico analysis. But we didn't want to stop there. We actually isolated RNA and DNA from pathogens and did not detect any bacteria that cause pneumonia, fungi that cause pneumonia, or viruses with our diagnostic tests. Neither did we detect from positive patient samples known to have other respiratory viruses. Our test did not pick those up. So again, we developed a very sensitive and a very specific diagnostic test. One of the things you do during the validation process is to see if you can detect patient samples with SARS-CoV-2. So we evaluated positive samples and detected 30 out of 30. That's a 100% positive percent agreement / sensitivity. And of course, we don't want to detect samples that do not have SARS-CoV-2. So we evaluated 30 negative samples, and we did not detect virus in any of those. And so March 13th comes around, the virus has spread to lots of different states. That's the day that Alabama reported its first case, and it was detected in 80 pH. By March 17th, we had our test up and running, so we went live with it. This is a copy of our sample report that we would give to providers. And we started with 150 tests per day. That was our max capacity. And we were doing this 7 days a week. To be able to inform the command center and infection prevention of all our results, we worked with Russell Griffin in medical school and infection prevention to establish a red cap system that would take our clinical data and report it to infection prevention command center. And then they could notify the nursing staff and medical staff when an individual tested positive for SARS-CoV-2 to get them into a negative pressure room and stop further spread of the virus within the hospital system. So this worked really well and continues to work, every hour on the hour since March 24th. Reports have gone to the command center and other individuals in the UAB system, informing them when there are positive results for SARS-CoV-2, with all types of graphics available to know if incidence rates are going high and low. It's broken up by employee and community and patients. So a fantastic database and fantastic thing that was set up to keep everybody informed, and so we could act on the results we're getting in the laboratory. So we provided 24-hour turnaround time. Most of the results were going out the same day, enabling providers to act on them quickly. And it really enabled UAB hospitals to return to operations. And so we were not only doing testing for UAB hospitals, but we were also doing testing for inpatients in other hospitals in our region. So this testing was very important because you could send testing to a reference lab, but at the time it was taking about a week, and that's too late to really act on it. What we focused on for UAB and for our community was providing 24-hour testing, rapid testing that these institutions could act on and really make an impact on mitigating the pandemic. So the VA, Medical West, Russell Medical Center, Grandview, Baptist, Legacy of Hope, and the Medical Examiner's Office, we were able to provide these results for them. Establishing testing allows you to know which patients are infected and which are not. And so that really allowed the start of clinical trials and the biospecimen repository here at UAB. And this is a picture of the amazing folks in the lab that made the initial SARS-CoV-2 testing happen. So a lot of work went into this, and I'm very proud of our team for making this happen. March 30th, ADPH confirmed the first 5 positive and 5 negative samples. It's something you needed to do to continue testing. And so our test passed this test. And then after that, it was really about expanding testing. So we started with about 150 tests per day. We purchased an instrument that allows us to do 60 extractions at a time. We got up to 200 tests per day. We purchased another instrument that did 48 at a time. That got us up to 300 tests per day. And then April 2nd, commercial diagnostic testing, which had been going to Washington and New York predominantly, now became available for UAB. So we validated the COBIS 6800 platform, and that got us up to about 700 tests per day. And so these are the amazing folks in the lab that made expansion of SARS-CoV-2 clinical testing happen. I'd like to point out Dr. Harada, Dr. McKinnon, and Gina as the leaders in the molecular pathology lab that got SARS-CoV-2 commercial testing up and running. And it was really quite a team effort, and I'm extremely proud of what we were able to do together as a unit. In the microbiology lab, we were able to implement Cepheid Expert PCR testing with a 1-hour turnaround time for the emergency room. We were able to establish multiple testing platforms to meet the needs of UAB. And so this is the Gene Expert Express SARS-CoV-2 test that was implemented in microbiology laboratory. I'd like to thank Bobby Vanderpool from Infectious Disease. She loaned us the instruments that allowed us to implement this testing and really give a rapid turnaround in the ED to be able to triage patients. So again, not just pathology, but infectious disease and the entire UAB community came together for this, which is absolutely superb. May 6th, within the Fungal Reference Laboratory, we validated other sample types. So bronchoalveolar lavage and cerebrospinal fluid were validated for SARS-CoV-2 testing. We also validated less invasive nasal swabs. So instead of the brain biopsy or the nasopharyngeal swab, that has to go all the way back in the nose, basically tickling the brain biopsy. We now had an option for a less invasive nasal swab, which goes about 1 inch into the each nostril. We have evaluated saliva samples, which we identified as too viscous to really work on the automated platforms. We thought it was difficult to handle, and we got about 85% sensitivity compared to nasopharyngeal swabs. So we abandoned this approach and stuck with nasal swabs. FDA was backlogged for a while, even though we submitted our EUA application in March. By June 23rd is when we actually got EUA approval for our test. And June 22nd, Governor Ivey announced an initiative to test 250,000 college students prior to return to school. And so we had to figure out how in the world to do this. It's an incredibly large number of tests. And so we got to thinking, and we thought pooling might be an acceptable approach for this. And so with some mathematics, we figured out with a 1.5% positivity rate, you can pool up to 8 samples, perform 1 test, and report 4 results. Here's a diagram of how this would work. We chose nasal swabs because they worked really well and 90% sensitive compared to nasopharyngeal swabs. So students would get a self-collected nasal swab. You'd have 8 samples. You take an aliquot from each, put it into one sample type. One tube, extract RNA just from that pooled sample, perform RT-PCR. And if it's negative, you can report all results as negative. So 8 negative results. That's wonderful. If it's positive, then you go back to each individual tube, test them individually, and report 8 individual results. And so we wanted to do this without impacting testing in the hospital system. The highest priority are inpatients, so that you don't spread the virus throughout the hospital system and ruin your healthcare system. And then patients showing up in the emergency room, we did not want to affect that at all. But we did want to provide this to keep the community safe and allow return to campus, to make it as safe as possible. So similar to March, we got on the phone, started talking to molecular companies like QIAGEN and Thermo Fisher, trying to see what was available, trying to get companies to be able to provide what we needed to make this happen. And we were fortunate enough to get 4 QIAsymphony instruments. These are large instruments that are high throughput that can extract nucleic acids.
Yes, I was looking for you.
We also were able to obtain 3 RT-PCR instruments to be able to help us with this initiative. And we were able to lock down an entire, it's probably 800 square feet room of extraction reagents for this initiative. So not without a lot of effort from a lot of people, particularly Jonathan Gidley in Hospital Labs and George Curavilla helping us to be able to do this in such a short period of time. And so we got the equipment, we got the reagents, and we made this happen. So we chose self-collected nasal swabs, 90% sensitive versus nasopharyngeal swabs. Saliva, there was a lot of news about saliva. It was the hottest thing out there. It worked for some places, but our in-house data proved it to be too difficult to work with in automated instruments. Oral saline rinse was evaluated, and that sensitivity wasn't good enough for us. So between June 22nd to July 1st, we received, installed, and validated reagents, instruments, and the pooling process. And just like submitting for our original test, we submitted our data for an FDA EUA pooling amendment. And so this is data showing that when you take one positive sample and add it to 7 negative samples, you can still detect it. And so 19 out of 20 positive pools with just one positive sample and a total of 8, we were able to detect 19 out of 20, 95%. So not bad. And we didn't detect any samples as positive if they were negative. So if there were 8 negative samples, there were no false positives in that group. So to make this happen, we really had to tap into our amazing community here at UAB, a research community, and also at SRI. And so we wrote this memo and distributed it out to the community, and the community responded big time. So we got lots of emails for interested individuals. We spoke with those individuals, try to see if they had the right skill sets, which were really aseptic and molecular skill sets. Recruited them, interviewed them, trained them, and scheduled personnel for a 24/7 operation. So we had over recruitment of over 20 individuals in the laboratory over this time. And at the peak, we had many more talented individuals coming to the laboratory to help with this initiative from hospital labs. So it's quite the operation in a short time frame. I'm extremely proud of the folks that did the work, and that we were able to get this done. We submitted the pooling amendment July 28th to the FDA. This is a picture of the individuals, not all inclusive, that help with the initiative. So we were able to result between July 28th to August 30th, up to 11,337 tests per day. Quite the amazing experience, and very proud of it. So we helped implement a safe return to college campuses, and the incidence rate during the surge was actually less than 1%. We detected PCR cycle thresholds in individual samples up to 39.9. And we were actually able, instead of doing one test and getting 4 results, we were able to do one test and get 5 results because the incidence rate was actually lower than the mathematical model I presented earlier. And the resources and infrastructure that we built was now used for campus sentinel and community testing. There's a picture of me a day before my birthday on Sunday night at 2 a.m. Helping with the initiative. We're pooling an all-nighter in the hood there. So lots of people contributed to this, and I think I've said I'm proud of this, so proud of this like 6 times now. So it was quite the experience. It's wonderful. From March to August, testing at UAB expanded from 150 tests per day to up to 11,000 tests per day. In the hospital system, we're at about 800 tests per day is what the demand is currently. From September 1st to present day, we've what we call the surge, with all the students returning to the fall semester. That ended, but campus sentinel testing continued. And so we continue to do that to this day. And just now, actually, there's a second surge for the spring semester to be able to test the students before they go back to campus. It's not as large as the return in fall, but still significant. So on September 17th, we took part in a FDA nationwide study to be able to see the limit of detection for all EUA approved SARS-CoV-2 diagnostic tests. And so when we received these spiked in samples in the laboratory, we evaluated them with our assay, and we got 180 copies per mil. Turns out that is the lowest limit of detection of any of the SARS-CoV-2 diagnostic tests, apart from PerkinElmer, which is tied with us. And there may be a couple more as this data continues to get published, but turns out with the nationwide study, we did develop the most sensitive diagnostic test for SARS-CoV-2. Here's a picture of Drew Freeman in the laboratory. Was instrumental in this work and the initiative and early initial testing as well. He's pointing to the CT curves here, the amplification curves, highlighting that our test was the most sensitive in the nation. And so with the flu season coming, one of the things we really needed to do was focus on flu. The big scare was that we would have a big flu season and patients would be coming down with flu and COVID and be significantly worse off. So what we did was modify our diagnostic tests to be able to incorporate primers and probes for influenza A and B. And so we did that. One test now gives you two results. You get a test for a result for SARS, and you get a result for influenza A and B. And so each target, flu and SARS, had very low limits of detection, 125 copies per mil. This is again confirming 125 copies per mil and 20 spiked in samples. Did not cross-react with respiratory pathogens, both bacteria, fungi, and viruses. And we were able to detect 100% of influenza positive samples and did not detect influenza in any of the negative known samples. And so we implemented this testing and it's helping with public health initiatives. Most recently, we've evaluated extraction-free PCR. So I told you about all these different instruments we brought in to extract RNA and then perform PCR. Do we really need them? Is the question that we're asking. And so both QIAGEN and a company called Biogx, which is local, developed extraction-free systems. This one is QIAGEN QIAprep and Amp Viral RNA kit. And what you do here is you have a primary sample. You transfer 50 microliters into a tube, and you heat it 70 degrees for 10 minutes. And then you take 8 microliters of that and put it into 2 microliters of proprietary solution, which is going to lyse the organism, get the nucleic acids out. And you add that 10 microliters to 10 microliters of RT-qPCR reaction mix. You perform RT-PCR, and you go from there. And so it's advertised that it's about 90-minute sample and answer. The reality, if you're doing a full play, it's more like 2.5 hours. But again, it bypasses all the extraction instruments that we spoke about earlier. So if you don't have these extraction instruments and you don't have reagents, then this is an option, potential option for you. And we'll see how well they perform compared to traditional testing. The Biogx system is similar. Here you rehydrate their reagent with 400 microliters of molecular grade water. You put 15 microliters into each well and then 5 microliters of patient sample. The magic happens during PCR. Nucleic acids come out and you get amplification and detection. This is advertised at 80 minutes, but similarly, if you have a whole bunch more samples, it takes a lot more time. And so this is a LOD table that you've probably gotten used to seeing by now. Our test would traditionally would be down here in about 150 copies per mil. Whereas the QIAGEN extraction-free system had 2,500 copies per mil as its LOD. So, significantly higher LOD means you're going to miss some samples with lower viral loads. The Biogx platform similarly had a LOD of 2,500 copies per mil. And so we evaluated a whole bunch of positive samples that we detected through the Roche Cobas platform, and also confirmed with the Fungal Reference Lab PCR test, which we developed. And what we see is 83.2% positive percent agreement. That's equivalent to sensitivity for the no extraction or the extraction-free QIAGEN assay compared to traditional RT-PCR, and negative percent agreement of 100%. So again, not bad. 83.2% is not bad at all. If you don't have any of the extraction instruments, you will miss about 17% of positives that traditional extraction systems and PCR would detect. But those are going to be samples with lower viral loads and those individuals may not be able to spread the virus as well as other individuals with a higher viral load. So if you don't have the systems available, this could be a good option. With the Biogx extraction-free protocol, the positive percent agreement from the data we have thus far is 85.8%. But we're expecting it to be more like 80% positive percent agreement because the sample set that we've evaluated thus far is skewed towards a higher viral load. And so in summary, early development of a diagnostic test enabled hospital operations to resume. Testing expanded from approximately 150 per day to up to 11,000 per day during the GuideSafe initiative. The pooling method helped enable a safe return to college campuses. SARS flu multiplex test is now guiding accurate therapy and public health measures. And extraction-free PCR enables rapid testing, but will miss low viral loads. So a little bit about non-COVID and diagnostic assays that we have available now or will come soon to UAB. So what we've been able to do during COVID is bring in a molecular viral respiratory panel that will detect a whole bunch of viruses and 2 bacteria that cause pneumonia on the same panel. It's a 90-minute test and works off of nasal swabs. We're also able to bring in a diagnostic test for tuberculosis that detects the organism, but also detects rifampin resistance. And we thought this was really important to be able to clear out negative pressure rooms to put patients with SARS-CoV-2 in those rooms. We implemented a new blood culture system called the Virtuo, which is able to incubate organisms and detect them faster. On average, for some organisms, about 10 hours faster to detection of the organism. We brought in new susceptibility testing, can now test for histoplasma in-house, and implemented a MALDI-TOF mass spectrometry system in the laboratory. Available soon using the same system, the GenMark ePlex. February 1st is our plan to go live with a system that once a blood culture turns positive, you can do a molecular test and be able to detect over 40 different pathogens and about 12 resistance genes that may be associated with those pathogens. So instead of having to wait 24 hours for an identification and another 24 hours at least for susceptibility testing, you can now be able to get an ID within 90 minutes of a positive blood culture and potentially data on molecular resistance, which is fantastic. We've also brought in fungal assays for pneumocystis and aspergillus, another one for aspergillus, and mold susceptibility testing we hope to implement here at UAB soon. These are the fantastic folks in microbiology and in the Fungal Reference Lab that have made this happen. So now we're going to switch and talk about some of the research we're doing. This is translational COVID-related research projects here. So we asked the question, can incorporation of prognostic markers into diagnostic tests enable optimized use of limited clinical resources? So to get at this question, we used the nasopharyngeal swabs and evaluated the cytokines that are present in these nasopharyngeal swabs to see if the presence or absence of certain cytokines predicted how an individual is going to do. So this flow here is very helpful on the logistics as to how this would potentially be utilized. So you have a symptomatic individual. They have a borderline clinical impression. So in the emergency room, you have to decide whether to discharge or admit this individual. You can't admit everybody, or every bed's going to be occupied, and then somebody who really needs it's not going to have it. So it's very helpful to provide this type of information. So we have the Sars test. I've put interferon beta here as an example, but this is a prognostic marker that would be in the multiplex PCR assay or the multiplex antigen assay. And so if you have a low viral titer, which you know based on CT values, and a high amount of interferon beta, which would be a good prognostic marker, then that individual is likely going to have a low risk of progression to severe disease. And that's the individual that's likely going to do better at home. So if you had to make the choice, that's the individual you're going to want to discharge home and save that negative pressure room for another individual. If there's high viral titer and low interferon beta or a low good prognostic marker, then that individual is going to have a high risk of progression to severe disease. And that's the individual you're going to want to admit and watch closely, because they're more likely to develop severe disease. So this is what we did here. We had 200 initial diagnostic nasopharyngeal swabs from SARS-CoV-2 positive patients. We performed the chart review. So mild was if there was no shortness of breath. Severe is if they were in the ICU, needed O2 support, or had fulminant disease and death. So kept it simple, mild versus severe. And what we did was take the remnant viral transport media after diagnostic testing was complete and quantified cytokine levels in patient samples using a flow antibody bead-based system, which is very commonly used in immunology, BioLegend Legendplex. And some of the data that we see is that for interferon beta, patients with mild disease express higher levels than patients with severe disease. Now, interferon beta is produced by infected cells. It then signals to the same cell to get them to be more resistant to viral infection, but also its neighboring cells. So it will bind to interferon beta receptor and cause them to be resistant to viral infection. So the more of this you can produce, the more resistant to viral infection you become. And so we saw with mild disease, individuals expressed a high level. With severe disease, they express significantly less. We also saw in severe disease less expression of RANTES and MIG. These are two cytokines that recruit T cells and NK cells. So at the start of infection, when you're getting diagnosed, if you're not making the cytokines that recruit T cells and NK cells, then you're likely going to not do well. That makes a lot of sense. You need NK cells to kill virally infected cells that with downregulated MHC1, and you need CD8 T cells to be able to kill viral infected cells. So if you're not activating these, you're not recruiting them, then you're likely going to do poorly. And so, patients with COVID, they tend to have a high systemic neutrophil to lymphocyte ratio. So neutrophils are very much activated in this disease. And what we see in severe disease is increased production of a CXCL5. It's also called ENA78, which is a neutrophil chemoattractant. If it's produced, neutrophils will go to that area. And so we see more of that in severe disease than in mild. And GM-CSF is an activator of neutrophils. It'll get them to produce reactive oxygen species, and its presence will allow for increased tissue damage. And so that's also increased compared in severe disease compared to mild disease. So we've identified these factors that are in the original diagnostic respiratory samples. And so ongoing studies are trying to incorporate these into diagnostic tests. We've also looked at the mRNA equivalent for viral infection. This is called subgenomic RNA. And so there's an issue where an individual tests positive for PCR during infection. And then, infection symptoms go away. The person's doing really well, but there's a persistently low level of viral detection in these individuals. That's called viral shedding. And what that does in the hospital system is that it keeps patients in the hospital. You don't want to discharge them because they continuously test positive by PCR. So we're working on a test to be able to detect subgenomic RNA, which is only present if a virus is actively replicating, to be able to distinguish that from the viral genome, which is present with viral shedding. So that's what we're working on here. And we've been able to target E subgenomic RNA and N subgenomic RNA. And some of the data with the E subgenomic RNA thus far indicates 20 out of 20 are positive percent agreement, and 20 out of 20 negative percent agreement. Providing diagnostic testing early on and having lots of samples come to our laboratory and with the microbiology background, it really was a ripe opportunity to collaborate with lots of people. So some of the collaborations that we have going on is looking at original diagnostic CT values, whether or not they predict patient outcome. We're looking at some PCR positive individuals that had high CT values that ultimately did not make systemic antibody. We're looking at the percent of Abbott Architect IgG positive samples that exhibit neutralizing antibodies. We're looking at microbiome and microbiome work in the original diagnostic samples and whether or not they predict outcome. And we're also working with a large 8-center, multi-center study trying to evaluate whether or not individuals with COVID infection have an increased risk of developing invasive fungal infections. So we know this entity exists. In some reports, is about 20% of patients with severe COVID are coming down with Aspergillus infections. Some of those reports are based on PCR alone. So we're doing a large study with Pete Pappas is driving this here at the UAB and other centers in the country to get the true incidence of invasive fungal infections during severe COVID. So that's the translational research we have that's COVID related. Some of the non-COVID research that we are doing is we're working on the development of a diagnostic test for C. difficile. One of the issues with C. diff is that 20 to 40% of individuals harbor it and will test positive by DNA test if they develop diarrheal illness for any reason at all. It doesn't necessarily mean it's because of C. diff. It's just you're carrying C. diff, you develop diarrhea, and you test positive by PCR targeting DNA. And there's another approach to detect it where we detect the toxin protein antigen. And that approach misses a lot of cases, as it's nowhere near as analytically sensitive as molecular tools. So we're taking a novel approach, which we think will work, and we'll find out hopefully by mid-year whether it can really get at that group of patients where they're testing positive for C. diff, getting treated for C. diff, but actually just have C. diff spores in their diarrheal illnesses because taking too much Miralax or Dulcolax or some other GI pathogen. So we're working on this test. Thomas Degemendi in the lab is really driving it. It's a collaboration with Martin Rodriguez and a former postdoc, Daniel Silberger, who is now in the Navy. Very proud of him. We're also evaluating the GenMark ePlex blood culture identification system. This work is in collaboration with Todd McCarty. It's really driven by Jeremy Meter in the Fungal Reference Lab. And this is a study to see whether incorporation of 90-minute diagnostic tests performed after a positive blood culture will lead to improved patient care, whether we can get individuals on the right antibiotic agents faster, whether this leads to earlier release from the hospital, the financial aspects of releasing patients from the hospital faster by getting them on better therapy. All these sorts of factors are being evaluated here, and we're doing both. We're doing gram positives, gram negatives, and fungi. So it's quite a big study. It's ongoing now. We're also working with other startup groups. So this is a company that came out of UAB. Some of you may know these individuals. Scott Barnum over in microbiology, and Teresa are the folks at C9 that we're working with. And this is a diagnostic test to be able to distinguish bacterial from viral meningitis, particularly important in pediatrics. And so this study is just starting now. We've also worked with other commercial diagnostic companies for fungal diagnostics. This is a simple dipstick assay for the detection of Aspergillus galactomannan. And we show using this system that if you just use a dipstick, you get 75% sensitivity compared to Platelia, which is not good. But if you use the dipstick and this cube reader, it's very simple to use, then you have equivalent performance of the two systems. So one system is an ELISA. It takes a while to do. It's complex. The other is a dipstick, which you can do at, we can't do this test at home, but it's the equivalent of like a pregnancy test, which is very simple to do. And so that's our translational research program. I just wanted to briefly talk about some of the mechanistic work, which we are also doing. This is focused on neutrophils and platelets during SARS-CoV-2 infection. And so this work is led by a graduate student, Sawanan Swatornian. She goes by Por. She's fantastic. And our postdoc, Mohammed Abdelgawad, also a fantastic individual. So they're really driving this work. So some of the autopsy studies in patients that die with SARS-CoV-2 infection were showing small vessel thrombi formation. We hypothesize that small vessel thrombi formation is being caused in part by excessive inflammation and activation of neutrophils in close proximity to endothelial cells, resulting in the release of collagen and platelet aggregation. And so we have the BioFlux system in our laboratory. This is a microfluidic system that flows blood across microfluidic channels, and you can image this over time. And you can label things like platelets and watch them adhere to collagen that's coated on the microfluidic chambers. And so this is what normal human blood looks like flowing through a microfluidic chamber. And white is fluorescent platelets that are adhering to collagen over time. And so this is what happens when you add factors that we show are upregulated in the respiratory tract of patients with severe COVID. You can see a lot more platelet aggregation than in the normal situation. And what we're doing in the laboratory now is trying to dissect out these pathways to see what's really involved. So I'm a mycologist at heart. There's lots of different molds you may have seen in your house or outside somewhere. I love these little critters. That's my background is in mycology and the study of the immune response to fungi, and also how the fungus resists, being killed. So fungal pathogenesis. One of my major career goals is to not let Dr. Moser down. This is Dr. Moser here. He was a director of microbiology and the fungal reference lab. We overlapped by about 1.5 years, and I learned a lot from him. And this is him handing me a key textbook in microbiology a couple of days before he completely retired. My other major career goal is to retire in style, like Steve. This is his, he's a big Porsche driver. And so this was his latest purchase. So that's 2 very good career goals. I'm going to introduce to you a neglected tropical disease called Philamentous fungal keratitis. This is mold infection of the cornea. It's a very straightforward disease. It occurs in immunocompetent individuals. You have mold spores. They get inoculated into the corneostroma, usually through vegetative matter. The spores grow into hyphae. And immune cells have to come from the peripheral lymph vessels to interact with the organism. By the time they get there, the spores are hyphae and they're very difficult to kill. And you get a lot of fungal mediated and host mediated tissue damage, which is what we are interested in studying and dissecting and hopefully coming out with therapeutics to help. So there's about 1.5 million cases worldwide, Philamentous fungal keratitis. And this is part of what we're studying in the laboratory. So very little known, limited treatment options. We established a mirroring model in the lab that enables evaluation of the host response and also fungal virulence factors. And what's nice about this model is you don't need immunosuppression, unlike other model systems. So you can really study the immune response to the organisms. There's a 2 to 5 day culture involved, depending on the mold that you're evaluating. This is Aspergillus fumigatus. You take the spores and you quantify them. And you use a syringe. You anesthetize a mouse. You abrade the cornea and inject 2 microliters into the stroma. Immediately, the corneal stroma turns opaque, so you know if you did a good job or not. And then you allow the disease to develop for 48 to 72 hours. And you track corneal opacity. We use fluorescent organisms so we can track the growth of the organism. And we do other techniques to study whatever endpoints we want to study. And so this is an example of the model system that we have. This is wild type mice and immunosuppressed mice. This is the level of disease that you normally get at 24 hours, 48 and 72 hours. We can visualize the fluorescent organism in the tissue over time. You can see if the mouse is immune suppressed, you get a lot more organism growth. As pathologists, we look at histopathology to evaluate the presence of the organisms and the immune cells that come in. And we also can quantify these measures and perform colony forming unit analyses. Another thing that's really nice about this model is we developed a neutrophil adoptive transfer model where you can take neutrophils from any mouse and inject them into the tail vein of a mouse. They will make their way into the cornea. Specifically in CD18 knockout mice, only the cells that you adoptively transfer will enter the cornea, and you can see wild type neutrophils from another mouse injected into the tail vein, entering the cornea and killing fungi. So you compare this amount of fungal growth to this amount. And then if you inject neutrophils that can't produce reactive oxygen species, you see that there's no killing involved. So really nice model systems that we've come up with in the laboratory to be able to evaluate neutrophil function. We also have a cell line in the laboratory, a mouse neutrophil cell line that we can genetically modify and study what we want to study. We have in vitro model systems where you take human neutrophils and you grow hyphae. Then you expose human neutrophils to the hyphae, and you can see they will bind to the hyphae, spread along the hyphae, try to kill the hyphae. The hyphae will try to resist being killed. And so we're studying this interface, what actually happens here, what receptors are involved, what signaling cascades get activated on both the host and the microbial side. And so the readout, after you allow this to occur for 12 hours, is how much fungus is present. If fungus just grows in RPMI media, this is how much fungal growth you get. If you expose human neutrophils, fungus gets killed. If you expose neutrophils that can't produce ROS, this is how much fungus is there. So it's really nice readouts. And so we're using these model systems to ask several questions. This is a fungal strain that lacks calcineurin. You can see there's no disease compared to the wild type. And so we are evaluating novel therapeutic agents, some of which exist currently to treat dry eye, to be able to inhibit fungal growth and the host response during tissue infection. We're doing dual transcriptome analyses to evaluate what's happening at this interface in both the neutrophil and the fungal level. We're studying fungal melanin, and we're studying antibody opsonization of hyphae during infection. So right now in the laboratory, we're doing lots of proof of concept studies to see what is worthy of further pursuit, and plan to invest time and effort in those pursuits. So one very interesting angle that our laboratory wants to take is, we're doing COVID-related research, we're doing mold-related research. Well, there is this entity, two entities actually, COVID and influenza-associated aspergillosis, where after or during COVID or influenza infection, the respiratory epithelial cells are damaged and you wind up with secondary infection. So a lot of these are bacterial secondary infections, but Philamentous fungal infections also occur. Like I said, up to 20% in some case reports, and we have a large study going on now in the US for 8 different centers to figure out the true incidence rates. And so we're very interested in how viral infection leads to invasive fungal infection in the lung, which is what we are studying. And we're developing lung models of invasive mold infection now in the laboratory. So in summary, diagnostics, early development of diagnostic tests really enabled clinical and research operations to resume at UAB hospitals and regional hospitals as well. Expansion of testing, pooling, and multiplexing continues to mitigate the infection. Translational research, we have a solid program evaluating commercial diagnostic tests. We're also developing our own and identifying SARS-CoV-2 prognostic markers. And mechanistically, we're studying COVID and molds, how they interact, and lots of different proof of concept hypotheses in the laboratory. So if anyone's interested in joining our program, don't hesitate to reach out. And there are lots of people to thank, both for the clinical work and current lab members, the Mycoplasma Lab, and funding. And I'd like to thank you for listening and for your time. And I'll take any questions if you got them.
That was fantastic, Dr. Leal. Truly amazing breadth and depth of your research program, and definitely a great way to start 2021. Thank you so much. It looks like we have a couple questions, a lot of praise in the chat section, but at least one question from Upender. Would you like to start?
Hello, okay. Can you hear me?
Yes.
Yes.
Sixto, congratulations. The outstanding contribution to this pandemic, you and your team. And I have 2 questions. How sensitive the PCR primers we are using at UAB in detecting the mutant variants? That's one question. And the second question is, I know in different parts of the country and in different parts of the world, different primer sets are used in PCR diagnostic testing. So whether these variations in the primer sets contributed to the extended pandemic.
For the first one, for the ones that are known, the UK and the South African variants, the variations are in the spike protein, and almost all the diagnostic tests have avoided the spike protein. They target nucleoprotein or the E gene. So all those are not affected, and fortunately, those are the ones we use at UAB and what most places are actually using. There's one diagnostic test from Thermo Fisher, which has multiple targets. One of them is the S, and it actually will detect the UK variant. So what happens with that test is all your targets are positive except the S, the spike gene target, which is negative, and that's called S dropout. And so that is a fantastic screen for the presence of the UK variant or mutations in the spike protein. What we've done is reach out to Baptist, who is actually using a Thermo Fisher assay, to see if they do identify any S dropout in the test that they're doing routinely. We've offered to be able to sequence it in our laboratory and to see if the UK variant is here. And Upender, I caught some of your second question, but not all of it. If you want to repeat it, I'm happy to answer it.
The second question is, when you review the type of PCR primers used in different parts of the country different institutions, as well as different countries are using different primer sets. This variability in detecting this by using this PCR testing, has it really contributed to this? Means false positive, false negatives might have contributed for this extended pandemic. What is your thoughts on it?
Yeah, there are a good amount of variability in different targets. I would say right now, with all the commercial diagnostic tests that are available, a lot of it has been standardized, and comparisons between different platforms, rarely does one platform detect another, detect the SARS, but another platform misses it. That doesn't happen too often. When it does happen, the most likely explanation for it is a limit of detection issue for the assay, which would happen when CT values are quite high, so very low viral load. That's most likely where the variability would be. I would say early on, it probably did happen when there wasn't that many positive samples available to really fully evaluate an assay. But right now, like the Roche Cobas, for example, most places have it, and there's thousands and thousands and thousands of tests that have been done on it, and variability with that platform between other platforms would have been identified, and I don't think is too big of an issue. So early on, yes, but I don't think right now where we're at, there's much variability.
I would like to make a comment. Sixto, you used the term "we were lucky" multiple times. I don't think you were lucky as much as you were a hard worker and you knew where to pursue. So we don't want any of the young people, residents, to think that luck has most to do with that, because it doesn't. There is a lot more that goes into being able to do the amazing amount of work you've done: connections, dedication, discipline, teamwork. So let's not think we are lucky that you are here and that you're part of our team, but luck was not the most important thing that led you to in this amazing journey, and has brought our department contributions to this level in the institution. I just wanted to remind you of that, as I'm sure you know. And thank you so much for everything.
I think the 2 a.m. the day before your birthday photo definitely highlights the work ethic that it takes beyond just a little bit of luck. So.
Yeah, exactly.
Definitely being in the right place with the right tools and the right work ethic.
Christina just wrote, luck is preparation meeting opportunities.
Yep.
Yes, yes. So everybody, and so you're an amazing role model for our residents. You had a great training, made great connections, and once you got here, you were ready to roll. And we needed you when you picked up and ran with it. So just unbelievable.
Thank you. I might go off camera. I don't want to cry.
Well, thank you again for leading us off in 2021. You definitely set the bar high for moving forward. That said, next Thursday, we'll have another UAB faculty member, this time from outside pathology, Carlos Arellano, who will talk about acute and long-lasting cardiac damage as a result of pneumococcal pneumonia, so another microbiologist. But thank you for all this. It was a fantastic presentation, and I hope everybody enjoys the rest of their week.
Let me remind you of the noon diversity talk
Yes.
that we have arranged. One more time. We'll see you at noon.
Bye, everybody.
Bye.
Thank you.